Hello my dear students, welcome back to my channel. In today's video, we will be discussing about the topic fish, which is a very important topic from the chapter Genomics, Proteomics and Bioinformatics of class 12 biotechnology. Before starting the video, I would like to request you all, if you are new to my channel, please subscribe to my channel for more such videos on class 12 biotechnology topics. So, first of all, what is FISH? FISH stands for Fluorescence In Situ Hybridization. Here the word fluorescence means the use of fluorescent probes and in situ refers to the exact site or the exact place of action, whereas hybridization refers to the complementary binding of the probe to the specific DNA fragment. So, what is FISH? FISH is a cytogenetic technique which is used to detect or locate a specific fragment of DNA. Means to say that a gene or a DNA fragment is to be located inside the cell. Therefore, the term cytogenetics, because cyto means cell and genetic means gene or DNA fragment, means a specific fragment of DNA is located inside the cell itself. Now, let us see the steps which are involved in this technique. So, the first and foremost step of this technique is the preparation of fluorescent probes. Now, these fluorescent probes are developed by the help of the technique which is known as Nick translation. A short video on this process, which is Nick translation, is already available on my YouTube channel. You can visit my channel and see the video on Nick translation for more details. So, after the probes have been prepared, the second step is the fixation of cells and its permeabilization. Now, cells on in which we have to locate the specific fragment is fixed onto a glass slide and is permeabilized. Permeabilized means the cell is made permeable so that our probe may enter the cell, because the probe has to bind to the DNA or the gene which is present inside the nucleus of the cell. So, for the probe to enter the nucleus or the DNA, the cell has to be permeabilized. Then the third step is the denaturation of DNA by a denaturing agent, that is formamide at 42 degree Celsius. Denaturation of DNA is done so that the probe may bind easily to the DNA. Then after the DNA is denatured, the probes are incubated with the cells, and during this incubation the probe may bind to the specific complementary location in the cell. If the probe has its complementary location inside the cell, it will bind. Otherwise, after incubation, when we will wash off the excess probe, the unbound probes will get washed off. And lastly, we will observe the cells under fluorescence microscope. And if our specific fragment is present in the cells, the probe would have bound to it and it will be visible, or the probe will fluoresce under the fluorescence microscope. So, this was all about the steps of the fish technique. Now, where is this fish technique applied? So, in your NCRT textbook, we have seen that this fish technique is used in the detection of CML patients. Apart from this, fish technique can also be used to study some chromosomal abnormalities. Now, what is CML and how the fish technique is applied in its detection? So, CML refers to chronic myelogenous leukemia, means it is a type of blood cancer, and in this disease, there occurs a translocation of chromosome number 9 and 22. This was found out with the help of a process which is known as karyotyping. Now, what is karyotyping? Karyotyping refers to the orderly arrangement of chromosomes. We all know that human genome consists of 23 pairs of chromosomes, and if we align all the chromosomes size wise, then the chromosome number 1 is the largest and the chromosome number 23 is the smallest. So, during karyotyping, it was found that in CML patients, the chromosome number 9 and chromosome number 22 had translocation. Translocation means that some part of the chromosome number 9 was transferred to the chromosome number 22, whereas some part of chromosome number 22 was transferred to the chromosome number 9. That is, both the chromosomes had some of their fragments translocated to each other, and this resulted in the formation of Philadelphia chromosome. So, previously, the severity of the disease was found by the karyotype analysis of the metaphase stage of cells. Metaphase stage of cells was used because during metaphase only the chromosomes are clearly visible, and as the blood smear of patient cells was used, therefore under normal compound microscope the scientists or the researchers used to count the number of translocated chromosome or the number of Philadelphia chromosome to find out the severity of the disease. Now, how does FISH technique helps in the detection of CML? Let us understand. So, with the help of FISH technique, the detection of CML would be very easy. How? Let us see. So this is a diagrammatic representation of the FISH technique where the fluorescently labeled DNA probe will bind to the denatured DNA, and after binding it will generate a fluorescent signal which will be visible under fluorescent microscope. So now let us see how the translocation occurs in case of CML patients. Now, as you already know that when the chromosomes are arranged size wise, then the chromosome number 9 would be longer as compared to the chromosome number 22. So here I have drawn the chromosome number 9 with red color and chromosome number 22 with blue color. Now in chromosome number 9 there is a gene called ABL gene, and in chromosome number 22 there is a gene known as BCR gene. Now during translocation, the lower part of the chromosome number 9 which contains the ABL gene breaks off and gets translocated to the chromosome number 22, whereas the upper part of the chromosome number 22 breaks off and gets joined towards the chromosome number 9. The shaded portion of chromosome number 9 with blue color is the portion of chromosome number 22. Now, due to this translocation, the genes ABL and BCR come close together. Now, this translocated chromosome in case of CML patients, this is also known as Philadelphia chromosome. Now, how this FISH technique is used in detection, let us see. So, what happens? So, from the DNA library, the clones containing the regions of chromosome number 9 and 22 were picked. Now, what is a DNA library? Like a library is a collection of books. In a similar way, DNA library is a collection of DNA. So, what happens in DNA library? Short fragments of DNA are inserted into vectors and the clones are kept like the books are arranged in the library. So, therefore, for making the probe for the CML detection, the clones containing the chromosome number 9 and 22 were picked for the preparation of probe. So, the DNA containing the chromosome number 9 region was labeled with red color or the red fluorophore, whereas the DNA containing the chromosome number 22 was labeled with green fluorophore. And when the smear of CML patients was taken, then after permeabilization and fixation of the cells, then these labeled probes were incubated with the cells, and after incubation the extra probes were washed off, and then the cell was viewed under the fluorescent microscopes, and in the patients where the translocation has occurred, then in those patients both the red as well as the green fluorescent probes bound at the same region, and due to this there occurs a yellow signal under the fluorescent microscope, whereas in the persons where no translocation has taken place, then the red colored probe will bind to chromosome number 9 and the green colored probe will bind to the region of chromosome number 22, and we will see clearly red and green signals. But in the patients where translocation has taken place, in those patients both the red and green probes will bind at the same region, and due to both of the probes binding at the same region, they will give the yellow fluorescent. So, by counting the number of yellow signals in the cells, we can see the severity of the disease in the CML patients. Now, this technique is also used to monitor the effect of drug or chemotherapy on CML patients. How this can be done? So, what happens that if we are to treat a CML patient with a drug, or the effect of chemotherapy is to be monitored in the patient, then before starting the treatment or before giving the drug to the patient, the smear of the cells of the patients will be made and the number of yellow cells will be counted. Then, after the chemotherapy or after the drug treatment, again, the blood smear of patients will be taken out and the number of yellow cells will be counted. If the number of yellow cells have decreased after the chemotherapy or the drug treatment, then we can say that the chemotherapy or the drug treatment is effective. Whereas if the number of yellow cells does not decrease, means the treatment is ineffective. This was all about the application of fish technique in the detection of CML. Hope you liked the video. Thank you for watching.